Hello students, I am Dr. Vinod Shinde. In the previous lectures, we have discussed about the tricarboxylic acid cycle and respiratory electron transport system along with EMP, HMP, ED and PKP, phosphoketolase pathway, pentose phosphoketolase pathway and hexose phosphoketolase pathway. Now, we will discuss about the ATP generation in these all pathways. In the glycolysis or EMP pathway, we have a ATP production by oxidation or substrate level oxidation is totally 4 ATPs were produced. In the sixth step, that is 1,3-diphosphoglycerate to 3-phosphoglycerate, and ninth step, that is phosphoenol pyruvate to pyruvate. The 4 ATP were produced, and ATP consumed were in the first two steps, that is glucose to glucose 6-phosphate and fructose 6-phosphate to fructose 1,6 bisphosphate. In these steps, 2 ATP molecules were utilized. In total, we have net gain of 4 ATP generation in the substrate level oxidation of ATP. In the ATP from hydrogens, that is from respiratory electron transport system or hydrogen transport system, we have all the way 6 ATPs are produced from 3-phosphoglycerate or glyceraldehyde 3-phosphate to 1,3-diphosphoglycerate, where 2 molecules of glyceraldehyde 3-phosphate and were utilized to produce 6 ATP molecules. In total, in glycolysis, we have 8 ATP molecules produced in the aerobic glycolysis. Now, if the glycolysis is further developed in the pyruvate oxidation, we have ATP generation in the pyruvate oxidation, that is pyruvate to acetyl-CoA synthesis, that is the first or precursor step of TCA cycle or Krebs cycle. Here 6 ATP molecules were produced. Then if it goes to the Krebs cycle or citric acid cycle, we have all the way around 24 ATP generation. In the seventh step, succinyl-CoA to succinate, we have 2 ATP molecules, that is actually GTP molecules produced, which are converted into ATP molecules. Then in the fourth step, isocitrate to oxalosuccinate, we have a NAD-NADS2 transformation, that is hydrogen transport, in which we have 6 ATP molecules generated. In the sixth step, that is alpha ketoglutarate to succinyl-CoA, NAD-NADS2 molecule formation, that is 6 ATPs were produced. Then in the succinate to fumarate, here FAD molecule, that is flavin adenine dinucleotide molecule was utilized apart from NAD molecule, and 4 ATP molecules were produced. Then the last step, that is malate to oxaloacetate, the 10th step, that is NAD acceptor is there. Here 6 molecules of ATP were produced, and in total 24 ATP molecules were produced in the Krebs cycle, and overall from glycolysis to Krebs cycle we have 38 ATP molecules produced. Actually 40 ATP molecules are produced, but the net gain is 38 because we have consumed 2 ATP molecules in the glycolysis, that is first step of glycolysis and third step of glycolysis, glucose to glucose 6-phosphate and fructose 6-phosphate to fructose 1,6 bisphosphate. This is the gross ATP generation, net ATP generation in the molecules. Now, in the next lecture, now we will discuss about the next point in the lecture is that bacterial electron transport system. How it is different from the eukaryote electron transport system. In most of the bacteria, respiratory chain is significantly different from mitochondrial chain, and difference between electron transport system in mitochondria and bacteria is as that electron transport chain in mitochondria is located in the inner membrane, while it is located in the plasma membrane in the bacteria. The similarity between two membranes has led to the proposal of the symbiotic theory of origin of mitochondria. According to this theory, primitive host cells which respirates aerobically by glycolysis were invaded by aerobically respiring bacteria like parasites. These invading organisms later came to live symbiotically in the host cell and become the mitochondria. The inner membrane of the mitochondria is presumed to have evolved from the plasma membrane of the invading organism. A closer examination, however, shows that the similarity between the respiratory chain of bacteria and mitochondria is largely superficial. Bacterial respiratory system have a great variety in their redox carrier patterns. One carrier may be replaced by another and there may be addition or deletion of specific carriers. Mitochondrial respiratory system are two-dimensional, linear and almost unbranched. Bacterial chains are often three-dimensional and branched, with entry and exit of electrons at many points. Electron may enter the chain through any of the several dehydrogenases and leave by any of the several oxidases. These are the terms that are observed in the bacterial electron transport system. We will discuss the fermentation or the fourth chapter, that is alcohol fermentation or fermentations, in the next lecture. Thank you.